Hello my budding biologists, and welcome back to exploring how cells coordinate activities within their internal environment and external environment. In this series so far, we've explored materials in how they move in and out of cells through the processes of diffusion, osmosis, active transport, and endocytosis and exocytosis. We've explored the factors that affect the exchange of materials across the cell membrane, such as surface area to volume ratio, concentration gradients, and characteristics of materials. We have looked at the cell requirements for energy, matter, and waste removal. And most recently, we have looked at the metabolic biochemical processes of photosynthesis and cellular respiration in meeting the needs of the cell. In this two-part series, we're going to be exploring enzymes and the way the environment impacts on enzyme activity. Enzymes are proteins found in cells that are responsible for speeding up chemical reactions. This is called catalyzing. Enzymes speed up chemical reactions to a couple of seconds that would otherwise take years to complete on their own accord. So enzymes are highly specific biological catalysts which increase the rate of metabolic reactions by lowering the activation energy. Let's unpack this so we understand it a little better. Enzymes lower activation energy of chemical reactions. Activation energy is the energy required to begin a chemical reaction between molecules. So this means in the presence of a suitable catalyst, for example an enzyme, the amount of energy required is lowered, meaning that the chemical reaction is facilitated more easily. As you can see in this diagram, the x-axis is the reaction progress, and the y-axis is the energy required to complete the reaction. The dotted line represents the reaction taking place without the catalyst enzyme. It requires significantly more energy to complete the reaction, whereas the bold line represents the reaction with the enzyme. This diagram shows the process of enzyme reaction at each stage of the reaction as well. So I have said enzymes are proteins. This means that they're a chain of amino acids known as a polypeptide that are folded into a specific shape. The way in which a protein is folded changes the final shape, which determines the shape of the active site. Okay, let's briefly review. Enzymes reduce activation energy of chemical reactions, allowing them to happen more easily and quickly. Enzymes are made of amino acid chains that are folded to make proteins and have specific active sites. Active sites vary in shape and reactivity. This makes them highly specific, and they only often act on one substrate. The substrate is the molecule that interacts with the active site of the enzyme and undergoes the chemical reaction. So enzymes are amino acid chains folded into proteins that have an active site. These active sites are highly specific for the type of chemical reactions that require less activation energy. Often enzymes are specific to one type of substrate. There are two main theories about how enzymes and substrates interact in catalyzing reactions. Firstly, there's the lock and key model. It uses the analogy that the enzyme is a key and the substrate is a lock. And the idea is that the lock substrate interacts with the key, which is the enzyme, and it suggests that the enzyme substrate have a perfect fit. The second model is the induced fit model. This model elaborates on the lock and key model, and it states that an enzyme's active site is not perfectly fitted for the substrate. However, the active site changes to accommodate the substrate to bind more effectively. In this clip, we've covered a lot of new concepts. Let me summarize them. Enzymes are biological catalysts that are highly specific to substrates. They allow for activation energy to be lowered and chemical reactions to occur more efficiently. Enzymes are made of amino acid chains that fold into proteins, and these proteins have an active site specific to the substrate. The two models to explain enzyme and substrate interaction are the lock and key model and the induced fit model. See you next time to learn more about how the environment affects enzyme activity.